Okay, I'd like to introduce our next speaker. It's Ken Oestreich, who is an assistant professor at the Virginia Technical Research Institute, as well as at the Biomedical Sciences and Pathobiology Department at the Virginia Regional College of Veterinary Medicine, which is here at Virginia Tech in Atlanta. He got his BS in chemistry from Tennessee Tech University, PhD from Vanderbilt University, so I'm assuming a Tennessee native.
Absolutely.
All right.
Not too far away from here.
And then he did postdoctorals at Emory University and the University of Washington. He's been at Virginia Tech relatively recently, since 2013. But even at this point in his career, he's getting some recognition. He got an Article of the Month designation for a paper he published in Nature Neurology, as well as a Faculty of 1000 designation for a paper that appeared in Journal of Experimental Medicine. The title of his talk today is CD4 Cell Differentiation in Experimental Theory.
So now for something a little different, disclaimer, I'm definitely not a computational biologist, very much a wet lab molecular immunologist. Our lab at VTCRI studies T helper cell differentiation, a topic you're going to be hearing a lot about today. And so I kind of just want to go through a basic overview and kind of historical perspective of T helper cell differentiation. Kind of a recent concept, the idea that you can actually have not only stable T helper cell populations, but also flexibility between one mature T helper cell type and another. The mechanisms, molecular mechanisms, that actually regulate those differentiation decisions. And then finally talk a little bit at the end about how computational modeling is a valuable tool to really comprehensively understand T helper cell differentiation. So I probably don't need this slide for this audience, but as many of you I'm sure know, the immune system is made up of a wide diversity of cells that come from both myeloid and lymphoid cell populations. This includes granulocytes, macrophages, B cells, natural killer cells, and the cells we're talking about today, T cells. Now T cells can be further subdivided into two flavors. CD8 positive cytotoxic T cell, which actually does most of the cell-mediated killing of the adaptive immune system. And then the CD4 T cells, or T helper cells, which do exactly that. They actually help to coordinate the adaptive immune response to a diverse number of pathogens. So it's actually been about 30 years since Mosman and Kaufman described the first two T helper cell populations. And what they described were two cells, a Th1 cell and a Th2 cell. And what they showed was a Th1 cell secreted a cytokine called interferon gamma, was actually responsible for combating intracellular pathogens like viruses. And also Th2 cell, which secreted a whole host of cytokines, interleukin 3, 4, 5, and 13. And these cells actually combated extracellular pathogens like helminthic parasites. And interestingly, these cells also seem to antagonize each other's both development and function. So we were actually stuck with two populations. A lot of great work was done trying to define these populations for about 20 years. But in the last 10 years, the last decade, there's actually been an explosion identifying numerous T helper cell types, including Treg, T regulatory cells, Th17 cells, T follicular helper cells, and more recently defined Th22 and Th9 cells. All of these immune cells arise from a single naive CD4 progenitor, and they all have discrete immune functions. So I just want to go through a few of the immune populations and talk about what their functions are, since you'll be hearing so much about these cells later on today. So I'll start with Th1 cells. Again, this is the cell that's responsible for coordinating immune responses against intracellular pathogens like viruses, certain bacteria, Mycobacterium tuberculosis, and Salmonella. They do this by secreting the very potent inflammatory cytokine interferon gamma, which then recruits CD8 T cells, activates macrophages, and promotes B cell class switching to the appropriate type of antibody for that immune response. Conversely, Th2 cells are responsible, again, for controlling extracellular parasites like helminthic worms. They do this by secreting interleukin 4, 5, and 13 to form mucosal barriers around these parasites, and also recruit eosinophils. Th17 cells, which I'm sure a lot of you have heard about, they've been the subject of intense scrutiny over the last few years in relationships to autoimmune disease, are responsible for responding against extracellular bacteria and fungi, including Staph aureus, Klebsiella pneumonia. They secrete IL-17, again, a very potent inflammatory cytokine. This also leads to the recruitment of neutrophils. Now, a cell type that's a little bit different is the T follicular helper cell. So this cell actually interacts with B cells in the germinal center of our spleens and lymph nodes, and actually kind of talks to the B cell and tells it what kind of infection is present and induces the appropriate type of antibodies or humoral-mediated immunity to combat those particular pathogens. And then finally, T regulatory cell, which is very different from the other 4 cell types, actually doesn't so much increase effector functions, but actually suppresses the effector T cell responses so that we can limit potential autoimmune disease caused by those cell types. And it does so by consuming inflammatory cytokines interleukin-2, and also secreting anti-inflammatory cytokines like interleukin-10 and TGF-beta. So a logical question to ask, and one that researchers have been looking at for a number of years now, is how can all these diverse T helper cell populations arise from a single naive CD4 positive T cell? And the answer is, as this cell encounters particular environments or cytokines, it actually upregulates particular lineage-defining transcription factors. You may also have heard these called master regulator transcription factors that actually go on to establish the gene expression profile of the cell. So for example, if this naive CD4 positive T cell encounters interleukin-12 and interferon gamma, it upregulates Tbet, and then Tbet goes on to establish the gene expression profile of the Th1 cell. So this occurs similarly with GATA3 for Th2 cells, FOXP3 for T regulatory cells, RR gamma T for Th17 cells, and finally the transcriptional repressor BCL6 for TFH cells. However, this is a relatively simplistic model. We have unidirectional development from a naive cell to a mature T cell state. This has actually come under fire lately by the identification that a number of T helper cell types can actually co-express lineage-defining transcription factors. So you can see all these papers here. There are a couple papers that are really more than a couple that suggest that Tbet and GATA3 can be co-expressed, that Tbet and BCL6 can be co-expressed, and then FOXP3 and a whole host of lineage-defining transcription factors can be co-expressed in these T helper cell types. And so what this has led to is a revision of the T helper cell model, where now it's not so simple where we have unidirectional development. We actually can have plasticity or flexibility between mature T helper cell types due to the co-expression of these lineage-defining transcription factors. So this has really led to a couple of questions. One, how can the co-expression of these factors be explained? So what's the driving force behind the co-expression of these factors in these cell types? And two, if you do have 2 lineage-defining transcription factors expressed in a single T helper cell, what's the functional outcome of that co-expression? So I want to talk about the first question here first. I should say at this point, I deliberately made this a little short. We're going to cover a lot of concepts, so please, if you have any questions, feel free to go ahead and ask during the presentation. So again, how can co-expression of these factors be explained? So I'm sure, as all of you know, our DNA is not just lying naked in our cells. It's actually a very highly compacted structure called chromatin. So what chromatin is actually DNA wrapped around a core of proteins called histones. And the internal tails of these histone proteins can actually be modified with particular residues, and certain covalent modifications are associated with gene activation, while other modifications are associated with gene compression. So for example, if you have trimethylation at lysine 27 of histone 3, this is actually associated with a gene that would be considered off. Conversely, if you have trimethylation at lysine 4 of histone 3, this gene would be considered on. So with that being said, one thing that researchers have done is actually to look at the epigenetic status of the lineage-defining factors, the loci, in these cells where these factors are being co-expressed, to determine what is the epigenetic status of those loci. And so these figures have kind of become commonplace in papers, but I just want to orient you to what we're looking at here. So we're actually looking at the gene locus of the Th1 lineage-defining transcription factor, Tbet. So it's called TBX21. And also the locus for GATA3, similarly named GATA3. We're actually looking at the histone 3 K4 trimethylation and histone 3 K27 trimethylation mark by chip-seq analysis. And again, the H3K4 trimethylation mark will be in blue, and the H3K27 trimethylation mark is in red. These are the cell types that they're looking at, so naive T cells, Th1, Th2, Th17, and then Treg populations. So not surprisingly, if we look at the TBX21 locus in Th1 cells, you can see a high degree of H3K4 trimethylation. Similarly, if we look at GATA3 in Th2 cells, again, you can see a high degree of H3K4 trimethylation. What was interesting was when we started to look at these loci in other T helper cell types. So in Th2s, and again, this cell type is very antagonistic to Th1 formation. In Th17s and in Treg populations, yes, you do see some H3K27 trimethylation, but you also see a fair amount, surprisingly, of H3K4 trimethylation. And this status is called a bivalent chromatin state, and it actually indicates that that gene is poised and able to be expressed in that cell type. If we look at the GATA3 locus, in all these cell types, again, you can see this bivalent chromatin structure where you have both H3K4 trimethylation as well as the H3K27 trimethylation. So what this indicated to researchers was that, in fact, you could potentially have flexibility between the Th1 and Th2 cell type because that locus remained in that bivalent chromatin state. Interestingly, if you look at a Th17 cell, you have a bivalent chromatin state at both the GATA3 and the Tvet locus. So theoretically, you could express both of those factors, leading to cellular flexibility between a Th17 cell and Th1 and a Th2 cell. And this wasn't limited to, yes?
I have a quick question based on what you showed
Sure.
earlier. Is there any point that a cell becomes terminally differentiated to one of those phenotypes in the sense that it cannot revert back to like a Th2 or a Th3, but it's committed to that?
Right. So in some cases, it does seem to be a unidirectional phenomenon. So it seems like a Th17 cell could only be flexible with the Th1 and the Th2. So we wouldn't necessarily predict that a Th1 could go to a Th17 cell. This is all going to be about environment. Because a little bit of what we're going to get into is you'll see that these lineage-defining transcription factors are going to respond to environmental signals. So presumably, if you didn't express a receptor for a certain cytokine, or that you weren't present in that particular cytokine environment, you wouldn't upregulate that factor. Therefore, you wouldn't be considered a quote-unquote stable population. However, it does seem you can overcome that type of T helper cell population if you are in that particular cytokine environment and can respond to it. So there are limitations, but these cells do seem to have a high degree of flexibility.
And that maintains throughout their lifespan.
Unless, so this is definitely a field of current investigation. So the question is there some other epigenetic mark that would come in and shut down that locus for good so that it could not respond? Sure.
How much are there effector functions related to this? I mean, we now have a cell with these bivalent chromatin activation sites.
Right.
These genes are shifted, they're active with lots of them. And we know if like a cell is essentially a P17/PH1.
Right. So in some cases it seems to create, and I'm going to go into this a little bit more when we're actually talking about the interactions between these lineage-defining factors, but in some cells it seems to actually promote a given individual cell type, so you have flexibility going from, let's say, a TFH cell to a Th1 cell or vice versa. In some cases, some people have reported you end up with a kind of a hybrid cell. So I'm going to talk about a cell in here in a minute that's called a Th2+1 cell, and that's a cell that actually can secrete both Th2 cytokines as well as the hallmark Th1 cytokine interfering gamma. Yes?
Curious how you select these cells.
Well, those particular experiments were actually done by KG Xiao's group at NIH, so not my group. I believe that they just went through standard purifications systems. Exactly, and then they did the choosing analysis with the antibodies. Okay. So again, this doesn't seem to be limited to the Tvet or the GATA3 locus. This is actually a very interesting dynamic between Th17 cells and Treg cells. What's interesting about this is these cells are very opposite. Th17 cells, very inflammatory, secreting IL-17. T-regulatory cells doing exactly the opposite. They're trying to shut down those effector functions. So you can imagine that if something goes awry with this process, this is how you end up with a lot of autoimmune disease states. But as you can see, the RR gamma T locus is very H3K4 trimethylated in both Th17 and Treg populations, again suggesting that there could be flexibility or plasticity between these two cell types. Again, this shows you that the ROR gain-of-feed locus is completely shut off in both Th1 and Th2 cells. And so again, that's why in this slide, it looks like the plasticity will be unidirectional from a Th17 cell to a Th1 or to a Th2 cell. And then finally, some lineage-defining transcription factors seem much more promiscuous than others. If you look at BCL6, which is the lineage-defining transcription factor for TFH cells, not surprisingly, if you look at TFH populations, you see a high degree of HTK4 trimethylation. But the surprising factor is every other population, naive, Th1, Th2, Th17, and Treg, even though it's not on here, have almost the same degree of H3K4 trimethylation, suggesting that they actually would share multi-directional plasticity amongst all T helper cell types. So just to summarize that question, at the epigenetic level, it looks like lineage-defining factors maintain the capacity to be expressed in multiple T helper cell subsets. So the factors can be co-expressed. What are the functional outcomes of this expression? So I have a few cartoons demonstrating, again, a few reports in the literature that describe what happens when you have co-expression of these lineage-defining transcription factors. So in early fate decisions, so just in the beginning of T helper cell differentiation, when that cell is trying to go towards a Th1 or Th2 fate, you can actually have Tbet and GATA3 co-expression. This was described in a paper from Maury Blunscher's lab all the way back in 2005, actually. And so what they showed was when Tbet and GATA3 were co-expressed in early cellular differentiation, Tbet actually physically interacted with GATA3 and prevented GATA3 from binding to the interleukin 4, interleukin 5, and interleukin 13 locus, thus skewing that cell towards Th1 development. However, if you have co-expression during late cellular development, you end up with this Th2 plus 1 hybrid cell, where GATA3 is actually able to bind to interleukin 4, interleukin 5, interleukin 13. So this is like first Th2 cell, but if that cell encounters a highly Th1 inflammatory environment, interferon gamma, interleukin 12, it actually can upregulate Tbet. Tbet then binds to the interferon gamma locus, and you have this dual secreting cell. So this would be an example of a kind of a codominant competition versus a bidirectional competition where you have one cell with one lineage-defining factor winning out over the other. And then finally, you can have a scenario, which I've termed a dominant competition, where two lineage-defining transcription factors actually interact in order to enforce the gene expression profile of a given T helper cell type. So in this case, Tbet can actually recruit the lineage-defining factor for TFH cells, BCL6, which is a transcriptional repressor, to actually turn off the gene expression profiles that will be associated with Th2 and Th17 development. It's actually, this interaction that my lab has been working on for a while now, and the one that I want to spend really the rest of the time talking about. Is there a question? Okay, so when I first started studying T helper cell differentiation, I actually was very intrigued by Th1 cells. And so a lot of work had gone into understanding how Tbet was actually binding and activating Th1 gene expression patterns. So we knew Tbet would bind to interferon gamma, would remodel chromatin structure, would recruit other transcription factors, and that would lead to the activation of Th1 gene expression patterns. But in addition to activating gene expression profiles, these lineage-defining factors also have to repress the gene expression profiles of other cell types. So you have to be able to turn off the Th2, Th17 gene profiles. So I wanted to understand how exactly does Tbet repress the gene expression programs of these alternative T helper cell types. So I'm going to summarize about 2 years of work with one slide. And basically what we were able to do was to show that Tbet was physically interacting with BCL6. So this is just a typical co-immunoprecipitation assay. And so we identified two classes of Tbet target genes in Th1 cells. The classical target, where Tbet would bind and activate pro-Th1 genes. But a second class, where Tbet would interact with BCL6 and recruit this transcriptional repressor, since Tbet has no transcriptional repressive capabilities on its own, to repress what we're calling anti-Th1 genes here, but really what they are is pro-Th2, pro-Th17 genes. And so this answers one question. We identified a novel mechanism by which Tbet was repressing gene expression. But about the same time we made that discovery, BCL6 was actually identified as the lineage-defining transcription factor for TFH cells. So we said, wait a second, now we have one of these scenarios where we have two lineage-defining factors being expressed in the same cell type. So if two lineage-defining factors are expressed simultaneously in one T helper cell, how is it that TBET is controlling BCL6's activity in this Th1 cell so that BCL6 doesn't start to establish the TFH profile? And so that was the question that we set out to answer. So I think I've already covered TBET, but just a little bit of background information. TBET is a classical transcriptional activator. It binds to DNA through this centrally located T-box DNA binding domain in a sequence-specific fashion. Once it binds, it remodels chromatin structure and can directly transactivate Th1 genes like interferon gamma, CXCR3. BCL6, like oxidly, is a classical transcriptional repressor. Has an internal BT-pos domain that it uses for its transcriptional capabilities. It has a C-terminal zinc finger DNA binding domain. And again, it uses this domain to bind DNA in a sequence-specific fashion. However, unlike TBET, once it binds, it actually represses gene expression. So our first insight into how TBET was regulating the activity of BCL6 in Th1 cells came through a structure-function analysis, looking at which domains of BCL6 were necessary for the interaction with TBET. So what we did was to simply make N-terminal truncations of the BT-pos domain and C-terminal truncations of the zinc finger C-terminal domain and figure out which domain was necessary for interaction with TBET. So you can see if we have wild-type BCL6 and wild-type TBET, we have a nice, strong, complex interaction. If we truncate the N-terminal BT-pos domain, the interaction is still present. However, if we truncate the C-terminal zinc finger DNA binding domain, now we lose complex interaction, suggesting that it's the C-terminal zinc finger DNA binding domain that's required for the interaction with TBET. Now, if we do the experiment from the other side and truncate domains of TBET, you can see if we truncate the internal domain, the interaction is fine. However, if we truncate the C-terminal domain of TBET, the interaction goes away. But importantly, the T-box DNA binding domain remains intact. So what we hypothesized was that during interaction between these proteins, TBET was actually binding through the C-terminal zinc finger DNA binding domain and masking it so that it could no longer bind to its own DNA binding sites. So in Th1 cells, what you have is a situation where TBET can unidirectionally recruit the transcriptional repressive capability of BCL6 to TBET DNA binding sites to turn off these pro-Th2, pro-Th17 genes, but it can also prevent BCL6 from binding to its own DNA binding sites and activating potentially the TFH profile. So these lineage-defining factors are very busy proteins in creating cellular development. So this is a snapshot or a cartoon of what we think is going on in Th1 development, where TBET has basically three main functions that we know of. One, it's able to directly bind to and transactivate classical Th1 genes like interferon gamma. Two, can actually interact with and recruit BCL6 to basically create a TBET that has these site-specific repressive capabilities, where it turns off these Th2 and Th17 genes. And then three, it indirectly represses the TFH profile by sequestering BCL6 away from BCL6's target genes, one of which is another transcriptional repressor called LIMP1. And now, this is a cartoon, but we've showed all this using molecular studies looking at chip assays, gene expression studies, and I'll show a little bit of data here in a second. But what we're able to show is when BCL6 could not bind to LIMP1, LIMP1 levels remained high in a Th1 cell and actually bound and repressed TFH genes like CXCR5, BTLA, and IL-6 receptor. But it's important to note that our hypothesis is dependent on the expression of TBET exceeding that of BCL6. So obviously this does occur in a Th1 cell. We've got a lot of TBET, we have some BCL6. But we hypothesized that if there was a way to actually increase BCL6 expression in Th1 cells, BCL6 could actually bypass this TBET-mediated control mechanism, be able to bind to its own target sites, and then create flexibility between the Th1 and TFH cell types. And so we wanted to test this, and we asked, are there any conditions that naturally induce BCL6 expression in Th1 cells? So this goes back to the idea of the cell has to be able to respond to a particular environment in order to upregulate that transcription factor, which would then lead to the flexibility of the cell type. And so one of the cytokines that was mentioned in the literature that may control BCL6 expression, at least somebody had looked at it in CD8 cells, was actually IL-2. So they showed that strong IL-2 signaling actually would inhibit BCL6 expression, while weak IL-2 signaling would actually promote BCL6 expression. So we decided to take our Th1 cells, expose them to different environmental IL-2 concentrations, and then see what happened to BCL6 expression. And what you can see is when we place these Th1 cells in low IL-2 environments, both at the transcript level and at the protein level, we had a stark increase in BCL6 expression. So indeed, it did seem that Th1 cells, or the BCL6 locus in Th1 cells, remained responsive to a lack of IL-2 signaling. But then the question was, could this increased amount of BCL6 bypass T-bet-mediated control, bind to and repress its target genes like this transcriptional repressor BLINK1? So we performed a chromatin immunoprecipitation assay where you can actually measure whether or not a factor is bound to a particular regulatory region. And so we looked at the promoter of the BLINK1 locus, PRDM1, and indeed in low IL-2 treated Th1 cells, you can see the increase in the amount of BCL6 that was bound. And this led to a corresponding decrease in BLINK1 expression at both the transcript and again at the protein levels. So finally, does this corresponding increase in BCL6 and decrease in BLINK1 lead to flexibility between Th1 and TFH cells? And indeed, the answer does seem to be yes, as we saw increased CXCR5, IL-6 receptor, and BTLA expression in these low IL-2 environmentally treated Th1 cells. So we actually have identified a regulatory switch composed of T-bet and BCL6 that remains responsive to IL-2 signaling, and that can actually promote the flexibility between the Th1 and TFH cell types. Now, what we ultimately would like to be able to do with data like this is to actually be able to determine the environmental factors that these T helper cells respond to, the transcriptional machinery downstream those environmental factors, so that we may actually have the potential to therapeutically manipulate the immune response. So for example, if we wanted to generate a potent tumor immunotherapy response, or a response against a chronic viral infection, we want more of a Th1 type response. So is it possible to give patients perhaps interleukin-2 or some other unknown environmental factor that would actually skew the immune system, at least temporarily, towards Th1 development? Conversely, if we want to improve vaccine efficacy, we'd actually want to promote a stronger TFH response. So is it possible to give blocking antibodies to IL-2 or to IL-2 receptor to patients to temporarily, again, push the immune response towards the TFH direction? And this is just one example. Clearly people are looking at autoimmune disease, looking at the balance between Th17 and Treg cells. Joseph's group has actually done a lot of nice work looking at the flexibility between Th17 and Treg cells.
Are there any mouse models for this? Is this amenable to mouse genetics and mouse experiments?
So actually at the same time that we were doing our mechanistic work, there was a study that came out where they were actually giving high doses of interleukin-2 to mice. And indeed that disrupted germinal center formation and TFH cell formation.
It'd be interesting to see if locking this in, if they have problems with parasitic infections or with allergies.
Absolutely, you might expect that if you blocked a Th2 response, for example, that might be a way to effectively treat asthma, right? Asthma is a very potent autoimmune disease caused by Th2 cells.
So, I think I saw it in the data presented, but I saw that T-bet expression is constant no matter the initiate with this concentration now.
Right, and so at the time, what we were doing is we were looking at, can we really totally reinforce the Th1 cell fate and still push it towards the TFH direction, right? So what we did is we kept those cells under the pressure of interleukin-12 and anti-IL-4, which would keep, you would imagine, T-bet expression constant.
Yeah.
Now, what I would think is, if we gave those cells, and we're actually doing these experiments right now, if we gave those cells, let's say we took away IL-2 and we decreased IL-12, I would think that eventually we would start to see the cell move farther towards the BCL6 TFH cell, and T-bet expression would decrease, and I think you would see a stronger TFH.
Yeah, because I would expect that somewhere downstream BCL6 itself could suppress, or it's a transcription suppressor.
In fact, it's been reported.
It
has been reported to bind to and repress the T-bet locus, which is interesting. It seems that, at least in this case, Th1 conditions seem dominant over that phenomenon, but you can imagine maybe inside of physiological development, system that's a way to turn on T minus pro.
I might have missed it, but what species was all your experiments done in?
So these were all done with, exactly, they were all done with mouse.
So does the same transcriptional regulation hold true in humans as well?
So we actually would love to get those experiments, and we're starting to move in that direction. Those experiments have not been done in humans so far. As you can imagine, it's a little more difficult.
So I'm sure you're familiar with the thing that shows that TH1 type TFH cells don't actually induce antibody production from B cells, or induce antibody producing T cells.
Right.
So that's one thing. The other thing is in human asthma, TH1 cells are upregulated, and modulation tends to happen in other ways. Within asthma have been documented asthma. And that there are lots of co-expressing human cells, IL-17 and interferon gamma, IL-4 and interferon gamma,
Right.
co-expressing at single cell level. But I am curious about when would you have a situation where you have a little IL-2 environment in here?
So there are two ways. So when would you have a situation where you have low IL-2? So IL-2 expression is increased during the height of an immune response, right? So you have a lot of IL-2 that's being secreted. However, as that immune response wanes, at that point you start to decrease IL-2. What you may see is a natural progression from a TH1 cell towards a TFH cell state as the immune response is waning. We're actually looking at some other aspects where BCL6 may be involved in memory development as well, as you would have decrease in IL-2. So I think that's where you would see IL-2 loss. Now, there is another way to make a cell not see IL-2, and that is if that cell downregulates its IL-2 receptor. And so it's possible to have a high IL-2 environment, but if you can't sense it, then you wouldn't necessarily be subject to this type of regulation.
There are people that have
Exactly, yeah. And there are cells, TFH cells, we even see it with ours, they start to downregulate IL-2 receptors, suggesting that they may be more stably moving in the TFH direction. Sure.
Yes, yeah, the idea is very good, but the problem is we face in the practical field is that T-help cell differentiation usually under in microenvironment. Sometimes interact with the dendritic cells and then develop. Because when you deliver the cytokine and you give an entire body for a high concentration, but actually the differential is not that easy because it depends on the microenvironment and contact with the dendritic cell. And I sense the certain cytokine, it will not diffuse to the other cells when you just focus in the cell is contact. It's very difficult. The idea is good, but in practical it's very difficult.
Well, I think you're absolutely right. Antigen is going to play a huge role. That trumps a lot of the cytokine signals, right? So when you get signal 1, signal 2. However, those cells are not always engaged to dendritic cells, and so I do think this is an effect that could occur post the height of an immune response, as that cell needs to kind of alter its phenotype in order to more perfectly fit that particular moment of the immune response. So, but that's an excellent point. Okay, so I hope what I've convinced you of today is that there are a number of factors that are influencing T helper cell differentiation. So one, the external cytokine environment. Obviously, we've talked about that extensively. Secondly, the internal transcription machinery that's downstream of that cytokine environment. And then finally, you can't just consider whether or not a factor is expressed. You have to consider possible interactions that may occur between those lineage-defining factors. So T helper cell differentiation is a very complex problem to understand. And so this complexity actually makes it an ideal problem to be solved by all the tools that we have available. So not just wet lab research at the bench, but also using a lot of the computational modeling techniques that you're going to be learning about over the next week. And so I actually borrowed this slide from Audrey, who's going to be giving the next talk. But basically, what you can see is if we can generate data using wet lab approaches, there are a number of endpoints here where we can actually generate in vitro and vivo data. We can actually throw that into computer models. go through predictions using computer software like Opasi and other programs that we're learning about, this actually comes up with then new testable hypotheses that we can take back to the bench. And the cycle will go on and on until we actually have a much more comprehensive understanding of T helper cell differentiation, which obviously is required with these kind of new ideas. We can actually have flexibility and plasticity between these mature T helper cell types, owing to all of the factors that I've talked about today. So with that, I just want to not trip over the podium. And just want to acknowledge my lab again at the Virginia Tech Berlin School of Medicine and Research Institute. I want to thank Joseph and the organizers here at VBI for inviting me, and also acknowledge the budding collaboration that the Oestreich lab and the Plastic Gummy lab have going. Amy and Sarah were involved in a lot of the data that I showed earlier in the presentation. So with that, I'll stop, and I think we have a little bit of time for questions if there are any. So, all right, thank you. It's not entirely surprising. I guess we had a few.
I just think if you use different strains for your experiment, differentiate mice like that.
Not at this point, no. These are actually just C57 black 6 mice. We have used some knockouts looking at T-MET knockouts, but again, those are on black 6. Yes?
So I-21 is a TFH product. So,
Interleukin 21?
yeah. So I've compared I-21 secretion between your TH1, TFH-like, and TFH cells.
At the transcript level, but not at the protein level. And actually gets back to a point that was made in the last discussion, that's something that really needs to be done to have a comprehensive understanding, is look at not only the gene expression level, but also at the protein level, and we're doing some of that work right now.
Yeah, so I had
Just one second, interleukin 21 is increased in the TFH cells by transcript.
But what about the TH1 and TFH-like cells?
So, so we have a TH1 population, and then we have a TFH-like population. The TH1 population does not, it does make some IL-21, but we definitely see more IL-21 transcript, again, in the TFH-like cell. I'm sorry.
Sorry. So I had a quick question. So you talked about a lot of complex interactions between these cells. Like, I'm not a biologist, so I wouldn't be able to, I don't know the nomenclature of the strains. But from a modeling perspective, what kind of model, you can develop? Like, would it be an agent-based model, or would it be an equation-based model?
So I think Audrey is going to address that in his next talk, and so I don't want to steal his thunder, but he's going to talk extensively about that. So, yes.
I have a question from modeling perspective as well. If you are working on a fairly new field, I do not know much about the immunology or experiments. The way I do it is after I have knife cells, it's knife CD4, it's either TH1 or TH2. I do not have the other subtypes. So how important it is to include these subtypes in a model? Do you have to invest? Do you think that's very important to look?
I think it depends on the question you're asking, and again, I really feel like I know exactly what Audrey's going to be talking about, because I think I know exactly what you're going to be talking about, and he's going to address a lot of those questions.
And just to quickly answer your question, I'll also touch on his question. For example, some things that we've done with Ken is use the CD4 T cell model that I will show you guys in a little bit. But then we can specifically ask questions to the model of, okay, if I differentiate this computational model into a TH1 or a TFH, and now I have a differential concentration of IL-2, how is this computational model going to react? So we can mimic the experimental data for calibration of the model, the data that Ken presented, but we're also then to tweak the model and play with the model to generate predictions and say, well, but look, you have been looking at BCL6, but our model is pointing out to these other transcription factors. And that way we can also target and go back to Ken and say, hey Ken, have you checked your ROR gamma T in this scenario? Because the model is predicting that it's being upregulated at this time point. So this is how the model, for example, would play in the story that Ken developed here.
I just have one question. So most of the data you've shown cover in vitro polarization.
That's correct.
So what about in terms of inflammatory disease model systems or infections? Have you seen the same dynamic transcriptional crosstalk between the systems?
So we haven't looked quite yet, but those studies are planned and hopefully will be conducted soon.
So the immunity paper that you referred to for the methylation studies, they've used, as you said, all in vitro cultures. But do you think the presence of all these transcription factors, the methylation of these transcription factors in other subpopulations might be contaminating?
I suppose you can never rule it out, but I don't think so. The last data set that I showed for TFH cells, that actually was from in vivo derived TFH cells, so it looks like, at least in that scenario, that data fits very well with the full data. All right, thanks again so much for your